Hello, we're continuing with life science and cell biology in a nutshell. We're going we're going through we were learning about cell division, so and how cells divide. So this is one graphic showing division of the cell and its cytoplasm, and then an interesting fluorescent picture. So this is showing the nucleus, genetic material inside, nuclear envelope, and inside is the nucleolus, where ribosomal subunits are made. Chromatin. So you have a nucleoplasm, which is like a cytoplasm with containing dissolved molecules like enzymes, etc. Then the nucleolus and the chromosomes are surrounded by a nucleoplasm functioning to cushion and protect the contents of the nucleus. Nucleoplasm also supports the nucleus by maintaining its shape, provides a medium by which materials like enzymes and nucleotides such as DNA and RNA subunits can be transported through the nucleus. Substances are exchanged between the cytoplasm and nucleoplasm through nuclear pores. Contained within the nucleus is a dense membrane-less structure composed of RNA and proteins called the nucleolus. The nucleolus contains nucleolar organizers, parts of chromosomes with the genes for ribosome synthesis on them. The nucleolus helps to synthesize ribosomes by transcribing and assembling ribosomal RNA units. These subunits join together, forming ribosomes during protein synthesis. The nucleus regulates the synthesis of proteins in the cytoplasm through the use of messenger RNA. mRNA is transcribed off the DNA. So these segments serve as template for protein production. So the mRNA is a template strand. It is produced in the nucleus, traveling to the cytoplasm through nuclear pores. So the mRNA leaves from the nuclear envelope through the pores to the cytoplasm, and there in the ribosomes, the tRNA molecule, transfer RNA molecule, helps with the translation process of the mRNA molecule into proteins. So in essence, you're turning DNA into proteins through this subsequence of events. So DNA to mRNA and mRNA into proteins by the use of helper tRNA molecules. So let's review what we learned previously. Two types of cellular multiplication. So asexual is when the parent cell that is replicating is identical to the offspring or the resulting cell. So this is mitosis. One example of this is budding of a small freshwater animal called hydra going through. It grows bulbs that develop into separate developing offspring. Note, in asexual or mitotic replication, before the parent cell divides, each parent cell's DNA replicates in the nucleus, making a copy of itself In order for the daughter cells to be exactly such as the parent cells, when the DNA is doubled, the two daughter cells result, and they will have the same number as the parent cells, and the same exact same DNA code as well. So all you're doing is duplicating the number of cells. You're not changing around the DNA codes or anything. So one whole set of DNA yields two whole sets after division. So this is mitosis. And each chromosome in the nucleus replicates itself right before mitosis, creating chromatids that are the identical sections of DNA linked before they split. Spindle fiber inside cell controls movement of chromatids as they line up across the middle of the cell before the chromatids divide evenly in the final step. It's important that chromatids move and split correctly with help of the spindle fiber and the aster. So, in order to ensure each daughter cell has a full set of identical genetic information in their own nuclei, that results from the cell division or mitosis. So again, the steps were that before the cell starts expanding and changing shape, all the replicated chromosomes inside the nucleus in prophase, so they are getting ready to come out as they will be pulled by the spindle fiber. Condensation of chromosomes occurs. So when you condense chromosomes, you have you replicate them and condense them. So then you end up with two sister chromatids, which are two identical copies of each chromosome joined by a DNA element called the centromere, which is a protein structure. Movement of centrosomes. At the start of prophase, there are two identical copies of each chromosome in the cell due to replication and interphase. Replicated centrosomes from interphase move apart toward opposite poles of the cell. So these centrosomes are made up of centrioles, which are protein subunits. So powered by centrosome-associated motor proteins. So I believe myosin is one of these. So formation of the mitotic spindle will eventually segregate the sister chromatids. The nucleoli begins to break down. So it begins to break down in prophase, resulting in the discontinuation of ribosomal subunit production. This indicates a redirection of cellular energy from general cellular metabolism to cellular division. In this picture, you're looking at prophase stage cell. So the nucleolus disappears, chromatin condenses into chromosomes, the separation of the centrosomes, and formation of the mitotic spindle. And this consists of, as we discussed earlier, prophase has G1 phase, where the cell increases its mass, synthesis phase where the DNA replicates, and the G2 phase, where more proteins are produced, where the mass of the cell keeps increasing in preparation for cell division. And in this microscope picture, you have mouse cells nuclei showing chromosomes condensed in red, the nuclear envelope in blue, microtubules, which help with the changing of the shape, and this is in green. So the cytoskeleton surrounding the nuclei, which will help in cell division. Thank you.